things. I posted this supplemental chapter I told you about the molecular dogma. Kind of a self-study. The idea there being is that I'm guessing from past classes, people have had this and they go, oh, why are we doing it again? Okay, well, great. I get it. On the other hand, if it is kind of foggy, you should do it again and look at it. So as you see, you have weeks, about a month to get this done. So the big danger is forgetting about it. But look over chapter 4. I have the slides on in this folder, and I also have a voiceover lecture. Hopefully it's all working. If not, let me know. I'll fix it. Wasn't any problems when I look at things, but that sometimes changes. And then when you're done with that, make sure that you go before the 27th. Go into the assignments. This is a new link that probably wasn't on Blackboard last week. Go in here and take the quiz. Okay, it's self-directed. It's not that long, and see how you do. So just a little bit of an incentive to go over things and remember what DNA polymerase III does and helicase and all those great things of one of the best stories in biology, the way we all work. Also up and running now. I haven't sent out an announcement, but I'm announcing it now with you guys. You have a week to go over a quiz for the first part of the first chapter, first information. What this is, just so you know, is that it's about a pool of about 20, I think it's just around 21 in this pool, and 10 questions come up randomly. So, if you're working on it, you may not get the same 10 questions as your friend. Okay, big deal, but you can sit there and complete it at one sitting. You have your notes, you have things in front of you, get it done, and see how that goes. I'll give you it's not going to be a complete immersion, a really big time consumption kind of thing, but it's one of those kind of teasers to kind of keep you moving, get kind of idea of questions I ask. Some may or may not be on the test. Probably a few will. There's so many things to ask. But that will help you when we get down to chapter 2, get something that for you guys, that kind of thing. Yes, sir.
Will we be able to see all the questions after we take it?
I don't know.
Just to have an idea of what's
I checked it so that you should see your answer and the correct answer.
I mean, you said there's 20 questions.
Oh, no, that doesn't come up. I think you get to see your other. Okay, well, good on that. So we are in, if you're following along, some of you still getting used to Blackboard, most of you it's old hand. We're in week 1, and we are looking at the cell structure, which this is a nice way to enter into the course, because we all have the feeling of what the cell is like. Okay, we've been learning that for years, probably, bits and pieces of it. But just like we tend to think that things that are common and easy are maybe not as important as the new special stuff, this is far from the truth here. The cell membrane is one of the defining factors of how cells work. They define what they are, but they also define how they work and how they interact. So we're going to not only look at the cell membrane, cell structure, how things get in and out of the cell. We're going to look at how the cell has different membranes in different groups. Talk about that. And we'll think about some of the implications of what it means to be a cell. I mean, we know on one level we're big mammals running around doing a whole bunch of cool things, but we come down to having these few trillion cells, quite a few trillion cells, that we talked about the other day, and yet all those cells have a lot of importance in our body collectively. So we know that the level of studying the cell is really important. The first part of this chapter opens up talking about microscopy, and if you want to kind of read through that or skim through it, that's fine. We cover microscopy more in lab. I think most of you are in lab. If you're not, you probably have been around microscopes enough to get the idea. Essentially, microscopes, like the science itself, have evolved a lot through time and have given us a real big insight whether we use light or electrons. We tend to think, when we think of cells and bacteria, perhaps, is something we may have learned in first year biology or in high school, and this is that. Well, cells have only a few shapes. Now over here I have some rods, kind of hard to see. This is through the eye of a microscope at 1000x. And they might be small circles. Well, we know they're not a circle. We're looking at them in 3 dimensions. They're spheres. We call those coccus. And we get an idea that we can stain them pretty in different colors. This is kind of the tip of the iceberg of things. Okay, so as we get to think about more of the cell, I like to think about this diagram here. Okay, this is they've blown up. An artist has helped us to blow up his drawing, an E. coli cell, and I put this in your brain, at least an image for your brain to consider early on, because a lot of times they say draw a cell, we draw a circle, if it's a eukaryotic cell, we draw a little nucleus in it, a few dots for ribosome. We think it's kind of this vacuous area when it's actually amazingly well engineered in looking back at it. What you see here is the cell membrane. We get that. We see that as a lipid bilayer. I'll look at that some more. We have this pulley system with an engine on it that drives a flagella. Okay, that's anchored into the cell membrane. It's a really important factor. We see that the membrane isn't just like a balloon, just this small, there's this membrane. We see there's even stuff out on the other side of it. We'll come to realize that the gram-negatives are really good at putting things outside of their cell, where the gram-positives are a little bit more conservative on that. And then we look inside of it, and this cytoplasm is just packed. Okay, here are our proteins and ribosomes out through here. Some are communicating, some are doing enzymatic breakdown. And we get in here, we can imagine, at least, even if it's not labeled, that we have the DNA, the histone-like proteins that we know make up our chromatin, right? H1, H2A, H2B, H3, and H4. For the bacteria, we'll have some just like that. We realize that the DNA just isn't this ball of yarn, that it all of a sudden magically makes more DNA and so forth. So we get an idea of that. I like to share that with you. So even though now we're going to break down parts of those things and talk about, we get this kind of gestalt idea that this is a really packed vesicle that has a lot of potential. Well, like I said, some of you heard before. If I had to ask you what are the shapes of bacteria, and we were doing this in lab the other day, people say, well, they're a coccus. Sometimes people will call them a sphere. That's fine with me. But we'll see that root word, coccus. We also have rods. Okay, and the coccus, the diameter is equal anyway you go across, right? It's a sphere. With a rod, we have one of the axes is longer than the width. The length is longer than the width. It can vary how big a rod is. And then we say, well, spirals, sometimes we call them spirillum. You'll realize that rods are called bacillus. Oftentimes you go, wait a minute, bacillus, isn't that a genus too? Yes. You got to keep an eye on when things are italicized and when it's a genus, when it's a shape. But we can handle that. That's not a big deal. Just keep our eyes open. The spiral can really vary from something that's almost like a comma, which we'll later on call a vibrio, to something that's truly a spiral, like a spirochete that can tunnel through your muscles and make you terribly ill. Fenning, just as an aside, is one of the best photomicroscopists. He retired some years ago. I don't know where he's at now, but he was able to do with black and white photography what we're still trying to do with digital these days. He was just a a master at doing that. I've always loved these photomicrographs. And so we have a few other ones. We have the spirochete, okay, which is like a spiral. It's the tightness of the spiral that's a big deal. We'll go on to learn that it has an internal flagella that helps it to do this little twisty thing. You know, wait a minute, flagellas, I thought those were little tails on bacteria. It gets interesting. Okay, we have some that will have stalks and a hyphae with them. When we talked about Gallinella, we might have wanted to talk about the external yarn prosthecae as a type of hyphae. We see this word as a descriptor with the fungus, but today we're talking about bacteria. Some of these words, of course, do overlap on these things. And one thing that we often overlook, a lot of us, because we deal with laboratory cultures, is that we don't play a lot with filaments. What a filament is, it can be done two different ways. As you look at it, a filament really is nothing more than like a filament, like a piece of thread. Okay, you get that, a filament, I get that. But it can be several different things. In some things like the actinomycetes that, like we did a gram stain off your teeth this morning, and you brushed your teeth, whatever, you got these long stringy bacteria inside of there. They're more filamentous, okay, they're like filaments. So just having a long body makes up a filament. But also, if we're to go out to Lake Springfield and get some cyanobacteria, some of them are filaments. They're long strands, and you pick it up, they're all slimy and yucky, right? And they're made up of individual cells sometimes that make up that filament. Okay, so, we'll just talk about when these things come up. If you really remember the three shapes and these extensions, that's good. The Michigan State, which has one of the best departments of microbial ecology, recognizes somewhere between 12 and 22 different shape morphotypes. Okay, it's not, this isn't a course for us to memorize, gangs and gangs of things. That's not the point. But my point is that bacteria show an incredible amount of diversity, even with their shape. Okay, and that they do have implications for their adaptation. But I get my arrows right this morning. So when you think about cell morphology, there's some things to realize that are important and some aren't. Really, when you get down to morphology, it doesn't help you in figuring out a lot of things very predictably. In fact, even getting down to DNA level sometimes, it's if you're just going by the 16S, it's hard to figure out which bacteria are doing what. Now you get down to gene level, genes have function. The 16S is really a structural gene. So we use it as a descriptor. If cell morphology is a consistent type of a characteristic, we can use it to key out different bacteria. So when we say, Bacillus subtilis, that I isolated from the soil this morning, is a rod, and it's always a rod that helps us. Okay, some things, like E. coli, we all know E. coli, our favorite one. Some of you have T-shirts saying, "Save the E. coli." You have posters in your room dedicated to E. coli. We love E. coli, right? It's just one of those things, right? We can't help ourselves, Anyway, with E. coli, we say, well, that is a gram-negative. That's not, may have some meaning for us, but it's a rod. Even some bacteria will change their shape throughout their life. And you'll hear this in lab over and over again. If you're going to characterize your bacteria, which those in lab will be doing, do it on a young culture. As they get older, they can change. Coli in its older age, which is a matter of a week, will actually become more football-shaped than rod and almost spherical, to the point where people are calling it a cocobacillus, and they'll even say that it's pleomorphic, and have many shapes. A lot of bacteria do that as they change their environment. They'll do it. So that's why we can use it as a characteristic. Sometimes we define when we look at that characteristic. Okay. Some of the implications for shape have to do with physics. Okay. One of the big defining properties of life is having to deal with diffusion. big deal, what is it? Maybe you don't agree with me, but what's diffusion either way? If you were to describe diffusion, you don't have to give me a definition off of Google, how would you explain it? Go ahead.
It's like stuff moving across the membrane.
Hmm?
Stuff moving across the membrane.
Stuff moving across the membrane, he's got a piece of it. Okay, do I need a membrane? If I have a can of Old Spice aftershave, right, straight out of the CVS drugstore right here, and I open it up, who's gonna smell it first? In this room. These poor people, okay, there was, oh, Old Spice, that reminds me of old men and cigars. That doesn't matter, it's gonna hit them first, who's gonna get it last? We sit around for an hour, you guys in the back are finding, okay, that's diffusion. Do I need a membrane? No. So what is happening with the Old Spice molecules? They're spreading from concentration, from high concentration to low concentration. Okay? Diffusion is really important. Diffusion, and the limit of diffusion limits life. Okay? We can't rely on diffusion only. Now, sometimes we do, but then sometimes we need to add something to it. We need to add energy to move things around faster, to transport things more quickly, we being our cells. Okay, so when we think about diffusion into a cell, if I put a cell and it's into water, and it's encountering a bunch of nutrients, okay, which is going to get the nutrients into the center of the cell more quickly, a big cell or a small cell? Small cell, no, one is because it's small, and there's less of the radius is smaller. What's the other factor? Surface area. The surface area to volume ratio is really, really important. We kind of take it for granted because we have big plumbing issues. We've got big veins going through our bodies and all kinds of tubes and things helping our tissues to do our thing. So being a single organism was generally an aquatic situation. The surface area to volume ratio is really important. It can also affect, and we'll have a slide on that in a second, we'll come back to that. It can also affect swimming and motility. Now, we all know if we're going to try to cover a distance, we're walking through air, it's going to take some effort, but it's going to take more of an effort to walk through water. And if we were swimming, water has resistance, not only because it's more viscous than air, but it has drag on us, okay? But if you factor up, and we don't do it here, in aquatic ecology we do the math a little bit more, but if we factor up the combination of lowering inertia, because there's less mass, and increasing the surface area, it's really like, for you, like you'd be trying to move through really thicker than molasses, like really thick glue kind of things. The comparison in the amount of drag on the cell. So the shape can sometimes help a bacterium when it's trying to move, or it's going to move very much. Some bacteria don't choose to swim fast with a little propeller called a flotilla. Some say, you know what, I'm just going to slither through, more like a snake. They do more gliding motility. But size and morphology is a big part of that. So let's talk about size a little bit. We did already. When we're talking about prokaryotes, we're saying that they're between 0.2 micrometers to some over 700. In a moment, I'll even show you something bigger than that. Okay, 700 micrometers, 0.7 of a millimeter. And, millimeters, not that big. That's a big bacterium. Okay, and I think everyone's got an idea of these two cells we have in the opening lectures. There shouldn't be too many surprises there. Look over that. You should be able to recognize most of those terms, I would think, on that when we return to them. So when we think about things being cultured, most of our stuff, now these are drawings made by Winogradsky in the 1800s, but most most of them are gonna fall between, the length we talked about, and not get quite up to 0.7. We rarely see bacteria that big, unless we're starting to deal with filaments, and then that's really on one axis. Okay. People always say, how can you tell when you get water from the lake? You know, what you're looking at. Well, you start looking at color, you look at the width. You know, a filament, the eukaryotic is always going to be wider, hyphae, or clear from fungus. Here's kind of got the prize winner. The last I looked is Epilopysium Fishelsonia. And these guys, I think, are found in the gut of a fish. Look, these things are huge. They're 0.2 to 0.7 millimeters. They're absolute big. So people, well, how can they still be a bacteria? They're small. You're wrong. Well, they do the chemistry. They look at the DNA. They look at the way the cell membrane is formed. You go, yep, they're bacteria. So my point is that there's a lot more to it than three little shapes, and there's reasons for the different adaptations. Being a symbiont gives you a lot of different options than being free living. Here's just our example talking about surface area to volume and why this is important to be small. Here we have a sphere with a radius of 1. Okay. If we just go through and calculate the surface area by 4 pi r squared, we have 12.6 square micrometers. And if we go through and do the volume by that well-known formula, we have 4.2. So we have a surface area to volume of 3. This means that there's a lot more surface area to volume than if we just up the R by 1 micrometer, go through the same calculations, our surface area to volume is 1.5. We make the bacteria bigger and bigger, you know, we start losing that surface area. Now at first you go, well who cares? Well, if you're just relying on diffusion to move materials in here, this has the advantage. Okay, so size does have the advantage. Does that mean being bigger is wrong, or there's not other adaptations and transport systems? Sure there is, but if we're just talking about the basic physics of things, there's an advantage that they're being small. One of the reasons, you know, most of your cells are fairly small, has to do with diffusion. You know, it's distance to the closest capillary. You know, it's distance to an alveoli or something. Still comes down to diffusion. Diffusion in life, that basic part of physics, really defines a lot of what we see and how adaptation works. Okay, so nothing too earth-shattering there. I think we could all do that. For those of you who worry about things, if I were to ask something on this, I would provide those formulas, although most of us probably have those committed at some level of our memory. But I am a big fan of, you know, you don't have to memorize everything in the world. There's too much in the world to memorize now, but, you know, can you pick up the pieces, take the tools, and make things? That's what I usually try to emphasize. Okay, well, how small can you be? We just saw that, you know, Epilopysium is big. You almost, it's almost a millimeter. You know, you get down to these things. How small can it be? It comes down to you have to have the basic elements of life. You have to have an inside and outside. So you need some kind of cell membrane. You got to have some ribosomes. You got to have some DNA, and you got to be able to have a metabolism. So when we get down to it, here's a picture of some mycoplasma. Okay, these things don't have great shape. When you come to mycoplasma, they are truly pleomorphic. Okay, because they don't have a cell wall. You'll find it strange. They turn out to be gram-positives, and they don't have a cell wall. Well, again, it's chemistry, but they're very pleomorphic. They're further distorted because this is an EM. There's a little bit of staining problem going on here. But these are nice. They're in proportion. They're closer in the field than this thing. What's this cell? This happens to be in a mammal. Red blood cell, very good. Yes, you we're used to the nice coin-shaped concave pictures that the artist had, but, in real life, things, do have a little bit more variation to them. So the mycoplasma gets down to the lower requirements, and you can start stripping it down. Okay, you gotta have ribosomes, you gotta be able to express genes, you gotta have genes. Okay, do you really need to express a lot of genes? Well, bacteria can get around that by being parasites, or symbionts, or whatever the relationship is. So maybe the ones that are out there adapting and working in the world needs a greater set of genes to degrade wood or whatever it is they do. We'll start to see, as in general, thing is that when we start seeing things that are parasitic or symbiotic, they start to give up some of their genes over time. Why have them? I'm safe. I'm safe and happy in this person's bloodstream, as happy as a bacterium gets, but, I don't need all those genes. You take away the ribosomes, you take away gene expression to any extent, you get down to the level of viruses. They can be smaller, but they're not metabolizing. They need a host. They need a true host as parasites. They do not metabolize. Okay, let's get into the cytoplasmic membrane. I would say of the things that are just amazing, that evolved and probably from self-assembly, is the cytoplasmic membrane, the membrane that goes around the cell. Thin structure surrounds the cell. Okay, got that. Highly selective permeable. So not everything out there can just diffuse in. Okay, and luckily, not everything inside, when you make a bunch of some kind of gene expression, leaks out. We know cells are kind of leaky. Our noses are always leaking something, or our eyeballs or something, but, it's fairly selective. Simple monomer for the cell membrane of a eukaryotic cell. Also of the bacteria. What do we have? We know that we have this connector molecule that makes up the, if you were just to have a simple triglyceride, right, you would have this connector molecule, okay, here. What's that molecule called? It's got a C in its primal form. It has a C, then it has an OH on each C or each carbon. That's called. You gotta review your freshman notes, okay? Glycerol. Okay, so glycerol, I call it connector molecule because that's what it does. Off of the OH, let's just start with the fatty acid. This fatty acid is a chain of carbons. It can be double bonds or single bond, depending upon those carbons. And depends upon the type of fatty acid it is. But at the end of it, there's a carboxylic acid. Now, if you're a little foggy on your functional groups, take a look at some of those. You should be familiar with hydroxyl, which are an alcohol. You should see a carboxylic acid, recognize it, an ether. Some of the other ones, I suppose an aldehyde and ketone wouldn't hurt, although we're not going to go into sugars as much in this class. But look at the functional groups that connect molecules. That's why we say organic is, we hope that you have it, but we're not going to be going really into like the Grignard reagent or anything like that. So we have an OH here off of the glycerol. We have a carboxylic acid, which, does this thing work? So if I have that OH off my carbon, here on glycerol, I lose water, I have a dehydration synthesis, and I connect it. I do it again with another fatty acid, and I get another molecule of water. In the simplest form, you may have learned there's another fatty acid here, and we put it on there, and we have this triglyceride. Okay, we know something about triglycerides, right? We know it's a fat molecule or an oil molecule. We know that it's an efficient way of storing what? Energy. Why? Why, if you're eating, eating too many Krispy Kreme donuts and fried chicken, and all of a sudden we start getting a little thicker, why does things go to fat? All of a sudden, I was trying to fight fat, some of us don't care, whatever. What's the deal with fat and energy? Why is that, what makes that such a good way of saving energy? Think about your metabolism work that you did in your early classes.
It has to do with the high concentrate, or it's very saturated, so all of the carbons and the hydrogens in per gram, so it's seven grams per minute or something like that.
You're getting in you're packing in a lot of reducing potential, okay? You're completely reducing those carbon atoms. And when you take fat, look at it the opposite way. When you take a triglyceride apart, you take a fatty acid, you feed it back in to aerobic metabolism, where does it enter? It enters, I don't know what you're saying, but it enters, I hope you're saying, it enters as that acetyl group, right? So 2 carbons on this is going to enter straight into the citric acid cycles. Who cares about glycolysis? There's a few things that get there, but citric acid, oh my gosh, I'm getting about 32 ATPs, depends upon if I have membranes to go apart. Whatever, I mean, a whole bunch of ATPs here, a whole bunch of ATPs here. So, as opposed to having a big glucose molecule, you can look at glucose. There's a lot of oxygen in there. It is not huge. It's a good battery. It's not the best battery. And you can get that battery and reduce it down with more hydrogens, feed it back in, so this forms acetyl-CoA. Now we can make a lot of ATP directly. We can get NADH, H reduced, and then feed that into the cytochrome transfer system and make a whole bunch of ATPs. So that's the deal. It's the size thing. Okay, it's really good. So we know that. That's metabolism. Fatty acid and metabolism. We'll come back to that. We might want to look at that again. It sounds like people haven't committed a lot of that yet to memory. But the thing here is structural. Now, this part is really nice. Now, maybe you know this, just want to make sure. Yep, that's a carbon, carbon. So we get it. What does the dash mean, the shorthand thing? I just want to make sure I'm still getting to know you guys. So the dash represents what? We know that the little peak is a carbon. What's the line? A bond. What kind of bond? Good, you're getting them now. How many electrons? 2, yeah. Good. And if we make 2 little dashes there, right, we know they don't have all the hydrogens in here because this guy was like, I'm drawing these things all day, I don't want to draw hydrogens. But we put 2 lines here, we do some hydrogens, we have a double bond, 2 covalent bonds are cool. Now, let's think more about this. By just knowing basic chemistry, we're going to get a big dose of understanding the membrane. This part over here has what kind of property? Hydrophilic or hydrophobic? 50% chance. How many people think hydrophilic? How many people think hydrophobic? That guy out there does too. Okay, so hydrophobic, good. What does that mean? Then it doesn't like water, okay? It's phobic to water. Why? This is the deal with memory. Why? She's right. But why? Answer your phone.
It's not bonding to water
It's
as
not
well?
bonded to water, okay? We're chipping away at it. A little more.
Stable.
Stable, good. Water's stable too, but this fatty. I'm thinking you're saying fatty acid. Ding, 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 ding. She put it together. Non-polar. What does she mean by that? There we go. We know how to describe it. Even I can't remember all the stuff that the chemist guys told me, but yeah, we know that water, because it's sharing hydrogen, it's got a slightly negative part where that part of the tetrahedron is exposed, and partially positive where the hydrogens are. We got it's polar magnets and make up water. Okay, this thing is putting the charge all around it. If it's not a hydrogen, it's a carbon, but the electrons are real happy. They're like, I'm gonna share the planet. Carbon, hydrogen, they're all around it, so they are non-polar. Okay, they hate water. Okay, it had to happen someday. We can't all love water. So we have two of those in there. We have a connector molecule, glycerol. Okay, we're almost home. Now, with the cell membrane, it kicks out one of the fatty acids, and it puts in a phosphate. Now, what do you think? Phosphate, polar, non-polar, what do you think? Bing, 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 yeah, big charge, okay, we got that. So it's gonna like to kind of snuggle up to the hydrogen parts of water, and water will make a semi-hydration sphere around it, but when it gets over here, I'm just gonna stick over here. And we know this in a simple form. We see phosphate. We know that in our cell membranes and in the membranes of bacteria, that there's actually a small carbon and amino faction on the outside of the B variable. The most common one is ethanolamine, but there's many different things that can be on a phosphate. But now, what do we have? We have an amphiphilic molecule. Okay. Not only is that fun to say over and over again as we skip home, enjoy looking back at today's lecture, but we realize that it has both properties of being hydrophobic and hydrophilic. I don't want to confuse people. I know it's saying phosphatidylethanol-amine. I think I have it here. That's what it is. And there's the ethanol. Okay, so that comes on there. That's the most common one. If we had to do a survey of the bacteria, we have it. Now, that's kind of nice, and it actually helps the different ones stack in the membranes differently. And it's a conversation for a different day, but we certainly gives it that polar head. Okay, so a little bit of I hope, review. Okay, if it all sounded like I don't remember hydrophilic, then open your book, read that part again. If you're still confused, come and talk to me. We'll talk about it. So artists show it different ways. Here's our glycerol, carbon in here being black, white is the hydrogen, and so forth. And then we have the polar group on the end. Yellow here, that's the ethanolamine, yellow being phosphate. And the way we usually draw it, in our class, is we'll make two tails, because it's a diglyceride now, two fatty acids on there, and then we'll have the hydrophilic polar head. Okay, so however you draw that, that's fine. One thing that does come into play is as we get, if you have a lot more hydrogen, it's all single bonds, it's a saturated fatty acid. If a double bond in there, it's unsaturated, and it tends to affect the packing. Okay, so you get different oils and fats and things like that. Some membranes do the same thing. Not only is it important for just the way that they make those fatty acids, but we'll find that thermophiles will have phospholipids that will pack better. They have to be more lard-like, more solid-like at regular low temperatures. So when they get in the high temperatures, they just don't melt away. So it becomes important, just this simple little architecture of these molecules. Okay, probably remember this part. So this is the beauty of things. Until the 1960s, people did not understand it. And then at that point, they started freezing cells, and they did what they called freeze etching. They would hit the cell with a blade so sharp that it would take glass, snap it, and that glass edge, they tap cells with it, and they'd etch open. And they started to see stuff like this when they put it under the electron microscope. That freeze etching was really important. And Avery and those guys started to figure it out a long time ago. You go, okay, cool. But what was the deal is people were still thinking the cell membrane was kind of like a balloon or saran wrap around the cell, and then they started saying, oh no, it's a bilayer. How can that be? And wasn't that far along, probably way before your time in most cases, but you start saying, well, it could be, if we know that they can take the soap, or they can distill out, extract using polar solvents, simple things like methanol and other things. You can pull out the lipid and go, well, there's a lot of lipid there, you get that, some phosphorus there, and they start putting it together. And so, in this case, the nonpolar and the nonpolar of the two bilayers are together. Like likes to be with like. So, this is excluding the maximum amount of water possible. And these things are polar. And most of the time, I know not all cells live in water, but in general, as we think about the story, these things will interact nicely with water, really. Okay, these guys, the cytoplasm, is that in or out? It doesn't matter, whichever one's in or out. The inside likes to interact with cytoplasm, so that's good. So now there's a natural structure. There's not only the element of self-assembly, but there's inherent stability to the cell membrane. Now, if we had a big cell membrane here, in fact, you can do it with soap. You put a drop of detergent on water, cover the whole surface until you add more soap, and then it makes what? Micelles, right? And it starts to go around to exclude even more water. But if you're just to put even a monolayer across a pool of water and touch it, it's going to be real flimsy with the water. The cell membrane was thought to be that way. People, when it was explained to me in grad school, they said, imagine a big pool of the cell membrane, and it really is mobile. It's quite easy for things by itself to move through it. And really imagine this sea of lipids that, there's a few ships sailing around. Those are the proteins sailing around this big sea of fatty acids and phosphates. So by itself, the cell membrane needs a little bit of help. Okay? You put cell membrane by itself, it's still going to explode. So in most cases, you have to have something with it. That's what makes the mycoplasma kind of interesting that they can pull it off. So what do? You start putting things into the cell membrane. Things for different reasons. Most of these things fall in the category of cholesterol-like things and protein-like things. So sterols and proteins. Okay, we can have proteins that span the cell membrane, so they're like channels. Now, they just don't let everybody in and out. Okay, but we'll return to that at the end of this first part of the lecture. We can have some that are just partially in the inside, the outside, but they tend to integrate into that. As you might imagine, those proteins, when they're folding, tend to put the more hydrophobic amino acids on one side so that they like to be in there. They don't necessarily have to bond everything. So that interaction of hydrophobic-hydrophilic does impose structure based on the environment. Other things are in their phosphate. On the head of the phospholipid has what charge? At neutral pH, at pH around 7. I'll give you a hint. It's either negative or positive. Okay, thank you. Okay, so we got that. So, this is interesting too. Divalent cations will see bridge. They bridge into the cell membrane. They help a little bit of stability. It's not at the point that they become calcified in concrete, but we'll see over and over again that divalent cations will pick up a negative charge on this molecule and one over here and kind of sit as a bridge. Sometimes iron will set up into two of those bridges, okay, the little crosses, but that's less described. Okay, so we see that. We see proteins, we see divalent cations. What else can we talk about? I'm coming to the sterol, so I got him. Membrane proteins bind substrates and process molecules. Okay, diffusion's good. Sometimes if you have the ability to grab something, you should, because you're a little bitty guy and the world's a big place, right? Sometimes we feel like that. Imagine being a vector. The inner surface cytoplasm membrane interacts with lot of different proteins and is involved with the metabolism. It's also shown now, it wasn't when I first started teaching this course, that there seems to be a cytoskeleton very similar in its early composition to what you have in eukaryotic cells. So they're finding more and more information about the cell membrane as we have more and more tools to look at it. So I think I covered this pretty much okay. I think I'm going to keep on moving. Here's our stabilizers. Okay, we know that sterols, have in general this kind of structure. 4 rings. They're very planar. Okay, cholesterol belongs to this family of things. But think of it as inserting like a stave or something that'll give rigidity. So in bacteria, I'm not going to ask you to draw this, okay, but if you see the word hapnoids, they are a cholesterol-like molecule that will get inserted to help stabilize the membrane, much like we have things like that in eukaryotic cells. You're like, well, why do you do that? Well, remember, if you've got this really big floating mass of phospholipids surrounding a cell, you just don't want to get an endless disharmonic wave going across. Having a little bit of stability is the first part of it. We're going to take that a little bit further with the bacteria. They're going to wrap a shell around that, except for some things like the mycoplasma. Most of them just can't make it without a shell around them. Some of our cells have it, some don't, depends on where they're found, right? Plants have cellulose. Cell walls are not by any means something new to us old biologists, right? Now, let's start changing things up a little bit, okay? Let me back up, oh no, it's still here. So when I talk about the eukarya, the type of cells you have, and bacteria, here's our format. We have our triglyceride, okay, so this just means C and H, a whole bunch of those here, okay. And when I take the carboxyl and I put a carbon on the other side of it, I have a ester bond. That's a little bit vague. Look up an ester bond. But we'll need to know because in bacteria and eukarya, we have this ester bond going on. That's just putting a carbon onto a carboxylic acid, or a carbon moiety, depends on what we have on that. With the archaea, and I think I have some ideas why biology has some ideas why. What they do in the archaea, that whole domain, they take it and they link with ethers. Okay, so why do they do the ethers? Ether's a little more stable. It can take a little bit more extreme than what people started to think about. The stability of that bond helps to stabilize that membrane a little bit more. But also then, that might have been it, back when Earth was hot and smelly, more so than it is now. And but it's one of those characteristics that radiated through the domain and stuck with it. So, ether bond, ester bond. We'll probably get into about 5 or 6 different cellular things that distinguish the bacteria from the archaea besides where they live. And so it does come down to the biochemical level. Okay. The other thing, as we consider the archaea, a domain that, if you feel a little uncomfortable about the archaea, that's fine. It's one of those things, really, there's a big bias against the prokaryotes, right? We all want to learn about furry little pandas, but, this stuff is important too. So, not only do they have that, but then when you get to this moiety, it's no longer just a simple carbon, single bond or double bond hydrogen. They have a repeating unit, and that unit is this thing, okay? It's this chain of 4 carbons with a side chain of a methyl group, and that's called an isoprene. I imagine iso because it's somewhat balanced with this. I don't know what a prene means. Maybe a chemist can help me out with what prene means in chemistry, but that is what that is called, a 5-carbon moiety that has a 4-carbon straight chain in it. This gives the archaea some different options, even in the fatty acid level. So, did I go over our time? Did we go to 9:15? Oh, good. I've been working on papers all night, so time has become kind of like this continuum. So, I'll sit and talk to you guys all day, okay? Don't make me go back to the typewriter. So, okay. Keyboard. So, we see one distinction here, second distinction here, okay? As you can imagine, I would say, well, what was that? I'm gonna ask you that. I'll probably say, can you write down and describe, let's say, 5 of the 7 characteristics we talked about in class that distinguish the archaea from the bacteria. I mean, 2 big domains, it seems like something we should talk about. It's like talking about the politics of the North and South before the Civil War. It's a big deal, right? Well, in the cell world, this is a big, and now we have 2 of those, okay? In your pocket. So let's take a little closer look on what this means. Here's our isoprene. Okay, I just have a few slides showing different ways. Okay, so they show it that way, they show it this way. We're pretty, they even show it that way, yeah. Okay, now, when you start putting 3 of the isoprenes together, in general, they'll call it a phantoneal group. Phantoneal group. So we can kind of check off where the isoprenes are by that methyl group. If we count it, this isn't a great thing. They're not really showing us the double bond. That's okay. I'm sorry that this artist left those up, but realize that those are repeating isoprene units. Okay, it's a shorthand. Okay, so now we kind of see this and go, well, what can I infer from there? I got that ether bond. That's kind of neat. That gives me rotation around there, but I think I had that for ester. It's not a big deal, so that's not a big thing. But we kind of get an idea that these things can pack a little bit better, okay? That side chain, since you're not a total spinning, there's a side chain to it. At first, well, is that going to be a big deal? Not a big deal. We'll keep our eye on that aspect. But now we can see that some, or I will tell you, and you will see, that the archaea, some just have, instead of a phospholipid, they'll have this fatty acid. It looks a lot like the phospholipid we just looked at. Okay, so that's good. Now, some of the other ones, don't worry about these names, everything has a name. Some of them will say, well, you know what? I'm going to set up that glycerol ether and that phantoneal group on one side of the membrane. I'm not going to put that little space in there. I'm going to go straight through, so we get this quadrophantoneal group on a diglycerol molecule. So, what are the implications to your things for that? You have lipid bilayers. What if those bilayers were continuous? What do you think that might do to this cell? Anything? Be business as usual? I don't care. What do you think? It can make it more rigid. Okay. Is that a good thing? Is that a bad thing? For you at 37-ish degrees, you don't want too much more rigidity. You want your cells to be nice and permeable and get stuff in and out and be the bright, wonderful people that you are. Okay. But if you start coming out of a thermal hot springs or something like that, having that stability, that rigidity, would affect your melting point. So at those temperatures, all of a sudden, the cell in the hot water, is still permeable and ready to deal with life. It probably is breathing sulfur instead of oxygen in its metabolism. It's dealing with, geysers are above 100 degrees of boiling because they have solutes in them. So they're dealing with this. They're going, just mitigate the geyser. That's not a big deal. So that adaptation can become important. Not all, I'll come back to you. I'm just going to finish this thought, I'll be right with you. So not just, some students get this from year to year. Some think, they all have that. No, it's variable. Some will have, just imagine there's a slot here. Some will have the bilayer phantoneal ether groups, and some will have the uni type of membrane. And
I was going to say that don't most archaeal cells live in harsher environments than other organisms anyway?
this is where it started. You're right. We started to think of the archaea as the extremophiles, okay, and extreme osmosis, halophiles. Having one of these things could help there. We know that the extreme thermophiles and hyperthermophiles have members of the archaea. Started turning out that's some of the real cold ones, but also the extreme, I think of it as extreme no oxygen places. The methanogens have it, they don't have the unimembrane as much. So yes, we started out thinking of the archaea as the extremophiles. Oh yeah, let's go to the moon, they're up there, they're extreme places, okay, they're crazy, they live in those places. As we start to look now though, we find that the archaea are very common. I wouldn't say commonplaces back here, but we find them everywhere. I've had undergrads like yourself come to the lab and strain them out of the Illinois River. They're, in the Crin archaeota and Ewing archaeota submarines. So it started out that way, just like everything, just like three shapes. Started out, we know more. If you think of them as extremophiles, that's good. Maybe you learned about them that way. We'll learn there's more to it. But that's a good way of thinking about why some of these things evolve. Okay, so here's my point on these guys. Some can have this dual layer, these are supposed to be the isoprenes, and some don't. They have the phosphate to make it polar, but they can make this one layer. So you got another adaptation. Now this is one of those adaptations because like some have and others have this. Okay, still, at least some of them have this, and we don't see that with the bacteria at all, to my knowledge. This is just re-emphasizing. I put stuff in so when you look at your notes, you have notes. All right, so we have the ether, glycerol diether here. We have the isoprene units. I think we've gone through all this. We have a biphenyl, the diglycerol tetraether, 1, 2, 3, 4. We talked about that. So I'm putting just a few more names in there so that we can talk about things together. Okay, hopefully not too many names to blow you away. Okay, I've been through this before, just showing that the membrane can be intact, working back to someone's early figures, so you can make some connections. So let's think about, I've been crowing, okay, some membrane, so what? Let's summarize this so what. Okay, rather than, I don't know what Matthew was talking about, he talks to me. Okay, permeability barrier prevents leakage and functions as a gateway to transport nutrients into and out of the cell. To have it inside, and distinguishing inside from an outside is localization. Localization is one way to get past randomness and entropy, which helps to support life. The membrane does that. Yeah, a little bit of fat, a little bit of phosphate, holy smokes. Very simple, but it did it. One could think that a micelle might have formed early in evolutionary history, and certain proteins got inside of it and started early metabolism. Nothing as complex as what you are now, but one can think that self-assembly, I can see that happening if I could get organic molecules. That's always a big if, but evolutionists can imagine that. 2, protein anchors, okay? Having something at the site, at the port, right, where you can get things interacting with the cell is really important. All right? Having it to be very uniform, and you can count on it for your cells, where it's happening, how much is happening, how much energy you can shunt into those activities makes it a big deal. So having protein anchors is cool. If it was just a saran wrap membrane, probably wouldn't work very well for the protein. As you know, proteins have ionic amino acids, positive or negative. They have basic and acidic, and some are more hydrophobic, right? People in biochemistry, does that sound about right? It is. So, the proteins are able to establish themselves and integrate. Good for us in that. This is a deal. Okay. Energy conservation, the site of generation and use of proton motive force. Proton motive force. How those proteins get out there? This happens in your cells too. It probably happens in your mitochondria. But how how is this happening? How are those protons getting pumped out? What's doing that? Tell me something about it. Tell me anything about it. Does this happen during glycolysis? No. Happened before the citric acid? No. After citric acid cycle? Yeah. What does an ATH have to do with it? I know you're embarrassed, you don't want to say.
Secure the proton,
Yeah, those are hydrogen opposites, it's the electron shuttle. Okay, so what you're doing, you're doing this in a dimembrane, a bimembrane organelle called the mitochondria, but with the bacteria, they don't have that. They only have one major membrane. We'll learn some alterations on that, but this kind of pump is happening with the cell membrane. This is why the next part of this chapter is so important. If you don't have a cell wall localizing this, sure you can pump this stuff up, but there's tons of stuff that wants to grab a proton, why not? If nothing else, diffusion is just gonna steal it away, right? Back down to the basic course that dictates life. In doing this, what do you set up? You set up this perfect battery, right? With the membrane separating charge, you have this battery, you get an ion potential, it gets so great, it wants to flood back in, and the cell says, wait a minute, you come back where I tell you, and it puts it through ATP synthase, you get enough stuff flowing, and I got enough energy to put a phosphate on an ADP, and now the cell's ready. But this, setting up this battery potential, this potential, this is like work potential. This is that energy potential kind of thing, a different way of thinking about it. This is huge. So the membrane, without that, you're not working with the mitochondria, which is a form of bacteria working. Just some things, the cell membrane is selective. Okay, the more ionic you are, the harder it is to get in and out without something transporting you. If you're water, not so bad. So, and we know that, we get dehydrated with the water, you feel good almost, fairly immediately. Things that are smaller and or, not terribly hydrophobic, can move through glucose, doesn't work that way. In fact, when we break down cellulose is glucose, the molecular cutoff for things that can move through the cell membrane is around 600 Daltons. When you're saying glycerol, we're saying around 100. So, even size is dictated by what can move through the membrane, even if transporters. And this is just talking about the importance of transporter systems. I'm pretty sure in other classes you guys get this in a greater degree, so I keep this part of the chapter very short, and I'm not gonna blindside you, I was gonna ask you a bunch more, so what's in the slides is, as far as we'll take this part of it, because I think in cell biology, you do more of it. But we know that by simple diffusion is good, but we've already talked about its limit. Okay? It's physics, it is going from concentration level, and there's nothing really powering it besides randomness. Okay, moving from high concentration, low concentration. We get a transporter system, of course we're going to get the rate of entry, or something's doing it, but it's nearly always due to cost, the transporter. You are putting a little battery on there to make this happen. It doesn't happen on its own. And so we have, in general, 3 different ways of transporting things. One, we can put a proton there to add a little energy. We can move a phosphate around. Phosphate's one of our best currencies. It's right in the middle, so it's easier to break bonds and help things move through. Or we can actually have a more complex system, the ABC, the ATP binding cassette that can come in and help the transporter move stuff through the cell. So the big take-home point here is transportation, to such a degree that outstrips diffusion takes energy. And why do we need that? Just like we need catalysis. These reactions that go, maybe you'll encounter the molecule. It's better if you can transport it in and get it there. And then once you have it there, you might get a reaction to go, but a catalyst is gonna make it happen in a time that's relevant to the cell's life, to that metabolism. And there's limits. You do it too fast, you melt the cell. I mean, it just can't happen all at once. That's why we have citric acid cycle. We burnt up glucose, we took a piece of paper and burnt it, that's sugar oxidizing, right? And the cell did that, you guys would melt. You gotta control it, and that's what the citric acid cycle is. Okay, so there's the three ways of moving things through, or three different types of that: uniporter, antiporter, symporter. Uniporter is one single thing, probably not as defined. Antiporter we see a lot of times is you move one thing in, another thing moves out, and then a symporter, two things moving in together, taking energy to do that. Just some examples: the lactic permease moving through lactose, that takes a little bit of energy, so that's a good symporter. Antiporter moving sodium out and the hydrogen in, sometimes to regulate osmosis, we see that as something, and a few other ones here. Again, be familiar with these names and they're associated with how the membrane works. There's a lot of examples. I don't need that you memorize phosphate, symporter, and all those different ones. These are examples. We're gonna have plenty of things to learn, okay? We're trying to keep it, somewhat digestible. Questions? Cell membrane cool? Maybe, maybe not. Lemke thinks it's cool. I'll nod my head. That's all right. We're gonna move in then to the next part, which is equally as fun. This is where we start. This is kind of like, Tai Chi. It's kind of like the yin and the yang. When at one point there was one thing, all of a sudden there's two. Okay, this is when we start talking about the cell walls of the prokaryotes. Okay. So now we're going from, the prokaryotes and the archaea have a cell membrane. It's a little different than the bacterial domain. Now we're going to go into the bacterial domain and say, okay, what are some major differences? This first major difference is so profound that you can detect it using a stain, the Gram stain, that chemist, biologist figured out that's so eloquent, to even this day, even though we have this jet propulsion kind of technology of doing DNA, we can do a simple color stain, a differential stain, and go, oh, that's one type of cell, and these guys over here are that type of cell. Because we can relate the stain to cell morphology. Okay, so we'll keep our eye on the stain idea, but it's going to come down to mapping what we're wrapping around the cell membrane, and that's going to have a lot to do. We're going to learn the structure, and then throughout the course of time, well, when you have this kind of cell, this really shoots the crap out of you because of this reason, but this antibiotic works because of this reason, and so, of course, we have to lay down the basis of it. So here we are in a nutshell to just get it all out there. Over here, we have a gram-negative cell. Okay, this is in wonderfully grainy black and white. Now, what do they both? And over here we have a gram-negative, gram-positive. Now, what do they share? How are they different? That's going to be our first question. They share. Well, they both have proteins in a bilayer. They both have a bilayer. Okay. Now, the one other thing they share that students get confused about is they both have this peptide sugar layer, peptidoglycan. We'll go into the how that's composed in a minute. Okay. The gram-positive has layer upon. Now this looks like a big brick. Okay, but really, if you think about the peptidoglycan, it's more like a fabric. There's lines of sugar that are connected by peptides, and then they're woven together on top of other layers of that. So it's a fabric on top of a fabric, with the fabric being the sugar backbone and a transpeptidation reaction that puts the sugars together. So that's what makes this up. Let's not worry about teichoic acid right now. Lipoteichoic, okay. Now, here, outside of the cell membrane that they both have is a layer of peptidoglycan, a peptide sugar. Let's talk about it. Sugar backbone, peptide linkage. All right. Outside of this is the neat thing with the gram negatives. They're all over the place because this opens options up for them. Here they have an outer membrane. Okay. Oh, they have an inner membrane and an outer membrane? Yes. Yes. Okay. Some people still are going, I don't get it. That's it. They have it. Now this membrane is pretty. We know that all the way around. Here's a phospholipid, and turn around, here's a phospholipid. With these guys, the outer membrane has half of it the same. This is a phospholipid layer here. We'll get better pictures from this, okay. This is phospholipid with sugar on top of it. Okay, but it has some pentahexoses and different sugar groups on it. Now, there's other layers that the gram negative can have. Okay. This is why they're so good at pasting onto your lung or grabbing a substrate and being able to starting to dissolve their exoenzymes, transporting things into this space. They used to call it space, we call it matrix now. There's enzymes in this space that can further degrade it and then put the stuff in here. So these things, so that's why the gram negatives, you find them in soil, you find them in your ears, you find them in your gut. They're really good at adapting. These guys are too, but you find these guys they like to be a little more resistant. They're not crows, they're not mouth, that kind of thing. Good. So let's take them apart. Now, I'm going to ask you on the first exam this question, or I'm going to ask you this request. I'm going to say, draw for me a gram-positive and a gram-negative cell, emphasizing the cell membrane and cell wall. Okay. I'm also going to make this request now, and I'll make it on a test. Make your diagram large enough so I can see, not that you have to make it big, make the parts that you're putting together large enough so that I can distinguish what the different layers and structures are. And I'll go on in the past, I've gone on, I list the structures I want you to put in the diagram. There you go, and I'm going to ask you a few other questions about it. But what I get is, and I'll tell you right now, and some of you go through this, even though I'm going to say, I'd rather not get this. Some of you, first off, you say, well, bacteria are small, so there's a cell membrane, and here's the cell wall, and here's this thing outside of it. And I literally will get something like that. Now, maybe you're shy, this is not the time, okay? So, then other people will just draw circles, that works. I'd rather, when you draw the cell membrane, you don't have to draw a million, little bitty things, but I'd rather, you at least show me an inset of, what's going on here, okay, as a possibility. Okay, and that's the same all the way around. But, because I know you guys are kind of smart, but the test by our system in this society is the way I check that I know you're smart. Okay, so just prove it to me. I'm smart. Here you go. I can do this. So I say that now to put it in your mind. So when you look at these things, you can ask. It's not a secret. So that will be an essay. There's 10 points there. Go home, draw it. Make a T-shirt, whatever. So the gram-positive. When we look at a photomicrograph of this thing, from electron microscopy, we see the cytoplasm, we see the nucleoid in here, and we see the cytoplasmic membrane. If you look close, when you get a good monitor on your iPads and stuff, you'll see the bilayer, and you see this thick, kind of sugar coating. Okay, it's kind of like a big chocolate M&M or something, because you got this peptidoglycan multi-layered here, and we see it again there. I'm going to do this. So when you draw something, it doesn't have to be this beautiful, but you get the idea. If you put in at least a few little diagrams showing you the phospholipid and the multi-layer thing, I get it, and you get it. And even if you had to memorize it, somewhere in your brain, you get it too, okay? So I'll end on this slide. Here's our memory. We know all about this. It's not ester linkages, or estrogen, and we have proteins going through there. Don't worry about teichoic acid yet, we'll come back to that. Here is our, there's two major sugars in the peptide, glycan and acetylcholine and acetyluronic acid. We'll look at those next time. And this shows you a little bit, but they come through and they stitch them together, so there's multi-layers of that fabric, that chemical fabric covering these cells. That's the gram positive. We're gonna pick up there and go through the gram negatives, have some fun with motility, and get the rest of this chapter on. Oh, if you're in lab, I'm sorry, I have a typo. We are doing the spread plate, the pour plate, and the street plate. The names are right. I wrote down 16, 17, 18. I think it is 15, 16, and 17. And look at the names. It's in your lab book. We'll talk more about if you're preparing for the quiz and stuff. I don't want to deceive things. Go! So, I haven't looked at it. Do you have any besides left turn? The big thing is the exam.